Okay. Well, it's an honor for me to introduce our speaker today, who is Dr. Patrick Boyle from Department of Bioengineering here at the University of Washington. He is a close collaborator with our electrophysiology groups, but I think they're out of conference. So I was nominated to introduce Patrick, and I know him because of my affiliations with bioengineering through my mentor, Mike Rainier. So Patrick is an assistant professor in bioengineering and joined the faculty here in 2018. He is originally from Canada and got his Bachelor's of Science in Computer Engineering at the University of Calgary, followed by a PhD in biomedical engineering, where he started, I guess, his interest in cardiac electrophysiology, where his PhD thesis was on the fertility system. He joined the Johns Hopkins University in 2011 as a postdoctoral fellow and then assistant research professor. He's quite accomplished and has 50 publications in a whole range of journals, from computational journals to journals that have more clinical relevance, such as Circulation, Surface JCI, and last year had a very exciting paper in Nature Biomedical Engineering, which I think some of you know he's going to talk about. I guess before giving away what he's going to talk about, he's this unique area of computational biology and using clinical data to help physicians. It's a very exciting new area, and we're excited to hear from him today about his exciting work here.
Okay, thank you, Fareed. So we'll get right to it. It's a pleasure to be here. Thank you to the committee for inviting me. I will say we're always happy in bioengineering to come over and present our findings, especially collaborative work with UW Medicine. I was also, as Erica can attest, able to fix the projector before my presentation, so this is why it's always nice to have an electrical engineer around, as the EP folks in the room can attest as well. So the talk is going to be in 3 parts. First, I'm going to start with an intro to how we model atrial and ventricular arrhythmias computationally, how we construct models and simulate electrophysiology in them. There's going to be a little bit of cardiac EP background, since I figured it's not an EP rounds. So forgive me, those of you who are well versed in the subject. Then I'm going to talk about how we can use these simulations to drive progress in the clinic. And this is going to both cover previous work from my time at Johns Hopkins, as well as unpublished data that I'm working on here with Nazim Akoum, who's away this morning. And then finally, I'm going to talk about how we can really go into hyperspace with simulations and explore terrain that it seems to be in the realm of science fiction, but in fact, with the use of computational simulations, we can look forward and see what type of therapies might be used 20, 30, 40 years from now. So to begin, why would we want to model or simulate the heart? Well, powerful computing resources are becoming more and more abundant. We have an incredibly robust supercomputing ecosystem here at UW. The supercomputer we use is called HIAC. And it's very large and very nice and inexpensive to use. And plus there's an emerging propensity for cloud-based supercomputing that anyone can access. And so we think that just, in the next 10 years, there's going to be an increasing democratization in terms of access to supercomputing at a low cost. Detailed mathematical models of biophysical systems, including the action potentials of cardiomyocytes, are widespread and relatively straightforward to implement. And there are huge success stories, of course, from other disciplines where the use of modeling and simulation can really drive progress. So, aerospace engineering, I certainly wouldn't set foot on an airplane that I knew hadn't been tested with finite element modeling. Structural engineering uses finite element analysis extensively in order to build better, safer buildings. And then in the biophysical realm, we have computational genomics, which has helped parse these massive volumes of omics data in order to establish mechanistic links between genotype and phenotype. There are things like computational neuroscience, which have helped sort of better understand the mechanisms, how the brain works as a calculator, and also how we can design things like better neuroprosthetics. And a timely example, computational epidemiology. This is from a paper published last year in Scientific Reports, but the same group is using this technology right now to model the spread of coronavirus and to anticipate, hotspots for potential infection. And of course, there's also my favorite, the lighter side of computational epidemiology, which is modeling, and this is a serious paper, I swear, from a Canadian university, the spread of Bieber fever. So keep an eye out for those. So our interest is in cardiac arrhythmia. This is an animation showing both the electrical and mechanical activation of the heart. It's my favorite animated GIF on Wikipedia. You can see, and this is the part that the EPs can tune out and check their phones. Cardiac excitation, of course, precedes contraction, and that results in the delivery of blood to the body. The ECG documents the principal electrical events, the depolarization of the atrial ventricles, followed by the repolarization of ventricles encoded in the T wave. If there's no coordinated excitation, then the heart stops pumping properly, there's no oxygen delivered to the body, and you have sudden cardiac death within minutes. So arrhythmia is any disruption of the heart's intrinsic rhythm regulation. And of course, some arrhythmias, I would emphasize to the non-EPs in the crowd, are much less lethal than others. When a good example is atrial fibrillation, which of course poses no acute threat to health and well-being, but it's highly correlated as a risk factor for stroke. And something I want to highlight up front, especially for the EPs in the room, this is for you, we sort of broadly classify arrhythmias into two categories when we're thinking about the mechanistic underpinnings and the biophysics. On the left here, we have reentrant self-perpetuation, where we have, for some reason, the formation of these vortices of self-sustaining activity that can drive rapid excitation of the heart and override the pacemaker. And on the right, we have a category of focal or triggered ectopy, here shown as an ectopic focus. So basically an aberrant pacemaker outside of the sinoatrial node that spontaneously triggers and drives the heart faster than its own pacemaker. From a computational standpoint, reentry is relatively straightforward to model at the organ scale. Triggered activity is extremely difficult to model at the organ scale. And so I'm mostly going to focus today on reentry, but we are moving towards developing better tools to capture the nonlinear dynamics at the cell scale that result in ectopic pacemaking and look at how that can manifest as organ scale arrhythmias. Okay, so let's talk about scale. Modeling electrophysiology is a huge multi-scale challenge. Here, on the left side of the slide, we have a model or a simulation of the excitation of rabbit ventricles driven by a model of the Purkinje system. This dates back to my PhD work. And the events there at the spatial scale are on the order of centimeters or millimeters. And the temporal scale that's relevant in terms of looking at these events is on the order of seconds. In contrast, at the cell scale, we have events like the upstroke of the action potential, and not shown here, but implicit in the ionic model, release of calcium through antigen receptors in the sarcoplasmic reticulum. And those are things that happen on the order of microseconds, and the spatial scales of cells, of course, are on the order of microns. So this makes for a complex and difficult finite element problem to solve. And I'm going to go through sort of the different scales, how we overcome that. So at the cell scale, this is a schematic representation that gives you an idea of how we approach the problem of simulating electrophysiology. In this schematic, you can see that we represent the different populations of ion channels in the cell. We have sodium ion channels, different types of potassium ion channels, pumps and exchangers. And then we have representations of things like the dyadic subspace between the T-tubule membrane and the sarcoplasmic reticulum, L-type calcium channels, and so on, and release from the sarcoplasmic reticulum through ryanodine receptors. So these can be abstracted as a circuit diagram, which can be represented by a system of ordinary differential equations at the cell scale that can represent membrane kinetics, leading to pretty realistic models of cardiac action potentials. And so if we take hundreds of thousands of copies of that specific model and then plug it into a tissue scale model, what we have is the main building block for simulating arrhythmia at the tissue scale. And so here, and I promise this is the most math you'll see on any of the rest of my slides, we solve a system of partial differential equations using finite element analysis to represent the electrophysiological excitation in tissue. This usually requires, well, it almost always requires numerical solvers, and for any problem of meaningful scale, it requires powerful supercomputing systems. So then at the organ scale, what we're really concerned with is reconstructing the geometry of individual human or animal hearts as acquired from MRI or other clinical imaging modalities. And what I want to stress here is that we're concerned with two things at the organ scale when we're interpreting these images. The first is the macroscopic organ geometry, which is pretty straightforward. It's not super difficult to segment and delineate the boundaries of the atria or ventricles. But in addition, we tend to use contrast-enhanced MRIs that highlight the spatial distribution of remodeled tissues. So in the case of the ventricles, we have scar surrounded by peri-infarct zone, which is here in yellow. And the kind of context of atria, we have fibrotic remodeling of the atrial, and both of those are recovered as a matter of course from late gadolinium enhancement MRI. Okay, so what I want to argue to you, and what I want you to take away from this section, is that models can represent electrophysiological complexity. So there's a pretty simple simulation here. It's two action potentials. The time scale between the stimuli is about a second. And what we know is that generally two stimuli means two action potentials. But if we shorten the coupling interval between those stimuli, what we're going to see is abbreviation of the action potential duration, weakening of the upstroke, the amplitude gets smaller, it's hard to see here, but the upstroke velocity would also be slower. And at its extreme, this can lead to complete suppression of action potential. Okay, and this has major consequences at the tissue scale. So when we stimulate at the bottom part of this square-shaped domain of cardiomyocytes, you're going to see propagation, and there's a region that's still refractory due to the ongoing action potential. But this tissue is recovered and ready to go again. So if we apply a critically timed in place second stimulus, we see the initiation of a reentrant driver. So this is going to be a concept that comes up again and again during this lecture. So I wanted to really drive home that it's critical that the models can reproduce this type of self-perpetuating excitation, which from a nonlinear dynamics perspective, I can tell you is not a trivial problem. And the reason why this is so essential is because in the previous toy example, we had the two ingredients for reentrant arrhythmia. So the substrate, in that case, was the state of the tissue after the first stimulus, and the trigger, the sort of spark that lights the fire, is the critically timed second stimulus. And what we know is that many different pathophysiological factors can give rise to both substrate and triggers. And what we think is that reentrant arrhythmias are quite complex, so simulations can help us understand how they work and reveal new potential treatment options. So the conceptual basis is that we have a healthy beating human heart. We add substrate from disease, genetics, aging, whatever, and then we add triggers. It could be atopic beats, stress, and so on, and then that gives rise to reentrant cardiac arrhythmia. Okay, so moving on to the big picture applications of what we can do with these simulations. The big thing that and this is where sort of the earliest applications of computational modeling and simulation came forward, can really drill down and identify mechanisms. So once we have a good model, we can use it to dissect and sort of infinitely probe the different aspects of the electrophysiological phenomena that are being represented by the computational models. We can then use those tools to develop novel therapeutic approaches, both either with drugs or in terms of new devices. On the clinical, moving towards the clinical application side, with image-based models, there have been many examples of stratifying patient risk. So taking images, running simulations in the model and saying this person is more likely to have an event in the next X number of days, weeks, months than another. And finally, the sort of culmination of decades worth of research was really using computational models and simulation as a direct planning tool for clinical catheter ablation procedures. And so for the remainder of the talk, I'm going to talk about 3 of these areas, and they're labeled here A, B, and C, 2A, 2B, and part 3. So here we go. What do we do? And specifically in the translational applications, I'm going to talk about atrial modeling and simulation. So of course, as many of you know, AFib is a huge problem worldwide. It's the most prevalent sustained arrhythmia, and it's expected to increase at least 2.5-fold in prevalence by the year 2050. Many patients tend to transition from a more clinically manageable paroxysmal form of the disease into persistent AFib, which is characterized by episodes that last a week or longer. Long-term success rates for catheter ablation are relatively low, on the order of 50%, although it depends how you measure it and who you ask. And one of the things that we noticed right away when we started studying this a few years ago is that the relationship between the fibrotic remodeling that can be extracted from LG-MRI, which is highlighted here in the inset, and the initiation and perpetuation of reentrant drivers, so rotors, just like what I was talking about in the introductory section, that relationship is really poorly understood. And we think computational models can wedge in there and be hugely helpful. So this is work from my time at Hopkins. The paper was published in 2016, and this sort of set off a chain of events that, like I said, culminated in the publication of our first pilot clinical trial, which I'll talk about at the end here of this section. So we take late gadolinium enhancement MRI, we segment the left and right atrial surfaces, and we incorporate fiber orientations in the atria that give rise to electrophysiological anisotropy, and we use an atlas of fiber orientation since we can't extract patient-specific fiber orientations using things like diffusion tensor MRI yet. People don't tend to like being in an MRI scanner for 8 to 15 hours. So once we have that, we essentially can delineate the regions of non-fibrotic myocardium, which are shown here in gray, and those have a relatively normal action potential, although we incorporate some changes to account for the effects of persistent or chronic atrial fibrillation. But the real important bit is that in the fibrotic regions, we incorporate a remodeled version of the action potential that has a longer refractory period. It has a weaker, slower upstroke, and all of this is to recapitulate experimentally documented evidence of what happens in the context of TGF-beta signaling in fibrotic myocytes. And so the whole key here was that we thought we're going to pace these models from 40, 50, 60 different sites that are spatially distributed in the atria. We're going to do what would be impossible or very difficult to do in the EP lab. We're going to pace from all over the atria and see whether the pacing can induce reentrant drivers. And what we observe is the initiation of these rotors. Here, the magenta dots, I think I've lost my pointer. There it is. So the magenta dots indicate the phase singularities of the reentrant drivers. And so we tend to talk about those as kind of the eye of the hurricane. So that's the center of the vortex, and it tends to meander along a relatively limited path on the order of a couple centimeters at most. And when we abstract this as an activation map, we can see that collapsing all of the locations of the phase singularities over time into one big sort of slug. That gives us an idea that for this individual, for whom the images were used to reconstruct the model, this particular area within the fibrotic substrate has all the necessary properties to initiate and sustain persistent rotor activity. Okay, and zooming out one step farther, the key insight is that each patient we found has a specific subset of regions like that that are significantly more likely to harbor reentrant drivers during AFib. And that's illustrated by this image, which shows the same phase singularities, but now superimposed on the map of fibrotic remodeling from the LG-MRI. In this case, we have 4 distinct sites. And so, our concept is that those are the targets that should be gone after outside of the pulmonary veins in order to ablate the propensity of the fibrotic substrate to sustain atrial fibrillation. Okay, and like I said, that culminated last year when we published in Nature Biomedical Engineering the OPTIMA study. Don't ask me how we found a way to shoehorn that acronym. It was a long day in New Orleans, and we were tired. This was a BHA meeting a few years ago, actually. So the idea here was to take MRI scans of 10 patients prior to their AF ablation. These are all persistent AFib patients. I put the table in here because I know it's a clinical crowd, and this is the kind of thing where I could get in trouble because I don't fully understand all of these things. But what we really cared about was that the majority of these patients had a large extent of fibrotic remodeling, according to LG-MRI. And what I do want to highlight is that there were a lot of comorbidities in this cohort, high risk of stroke in many of them, and for the majority were not first-time catheter ablations. So many of them had multiple failed procedures prior to being enrolled in this pilot study. And what the clinicians told us was that, yeah, for example, patient 3, patient 9, multiple failed catheter ablations. The next step here would have probably been catheter ablation of the atrioventricular node and implantation of a pacemaker. So these are kind of the sickest of the sick who weren't eligible to be enrolled in any of the other trials that were going on at Hopkins at that time. And so here's an example. This was our 5th patient. It was in January 2017. The substrate stress test revealed multiple potential targets. Here's an example of one of them in the anterior left atrium. And here's what the clinician sees on the day of the procedure. This gets loaded into the CARTO navigation system and merged with the electroanatomic map that's built up on the fly. This is an actual photo from my phone of the screen of the CARTO workstation in the EP lab, where I was sitting there like kind of freaking out, worried about, is this actually going to work? Did we get the files to load this time? Did they update the OS on the CARTO workstation, which are the problems that we were dealing with during this study? But sure enough, when they went in, they were able to trace, navigate the catheter to the precise locations that we had recommended and deliver ablation. And this is cool. Like what we started to see was, this is a patient who's in AFib prior to the ablation of that particular target that I showed you, the left atrial anterior wall. And upon ablation, the patient converted to atrial flutter, which is a more organized rhythm. Now, this isn't the be-all, end-all, and we know that acute termination of AFib during an ablation is not necessarily an indicator of long-term success, but we figured that this meant we were on the right track. One thing that I'll really emphasize is that we took an extra step beyond just doing the substrate stress test. What we would do computationally for each individual is we would do one round of substrate stress tests to identify a set of targets, and then we would virtually ablate those and rinse, lather, repeat until the model is completely non-inducible. And that came up several times during the course of the study. So here's our 9th patient from June 2017. Initially, we only saw macroscopic reentry around the annulus of the mitral valve. And so our solution was to virtually ablate that and see whether there were any potential other regions in the substrate that could sustain reentrant drivers in the absence of that macro reentry. And sure enough, what we saw was that these gold regions represent virtual ablations. And what we see here is for initiation of reentrant drivers in the posterior inferior left atrium, which were previously concealed by the fact that the model was so much more likely to go into the macro reentry state. There's a different reentrant driver closer to the right side of the left atrium. Here's the patient-specific ablation plan. Here's the sort of checkmark shaped ablation pattern that was applied to knock out both of the targets. And this was Dr. Calkins doing the ablation. So always entertaining in the EP lab, lots of loud music playing through the stereo. And we saw termination of AFib, which was super, super exciting. And sure enough, when Calkins came around and tried to re-induce ablation, so pacing aggressively from the coronary sinus in order to try and re-induce AFib after the acute termination, we found that it was completely non-inducible. And so this was one of our most exciting moments of outright success. So overall, there was no recurrence of persistent AFib. Only one patient had a AFib slash atrial flutter at the end of the follow-up, but it was paroxysmal AFib that came and went. And right now there's a prospective trial planning underway at Hopkins in order to do this in a randomized manner. So the takeaways here are that we had a proof of concept for a completely new treatment paradigm. in atrial fibrillation ablation. We overcame huge technical and logistical challenge and emotional hardship, I would say, from the engineer's perspective. We're cautiously optimistic that the clinical outcomes are pretty good. And the RCT is enrolling soon. And I'm sort of still an adjunct participant in that from a technical consultant standpoint as part of a UO1 award that I'm a co-I on with Natalia Trianova. That's my mentor from Hopkins. So the next part of the presentation is going to be about work done here at UW with Dr. Acum predominantly in cardiac EP, and it concerns ESUS, or embolic stroke of unknown significance, which I sort of lump in together with something, a concept of cryptogenic stroke. And so this is patients who have a stroke but have no atrial arrhythmia whatsoever. And the interesting finding that Nazem published last year in neurology, this paper highlighted in the right-hand side, is that ESUS patients have just as much fibrosis as AFib patients, but of course they have no atrial arrhythmia. And that's an unexpected finding. So I thought I would put up Nazem's picture since he's not here. And Savannah Bafolco is the PhD student who's done this work. She's sitting in the audience today. So how can we use modeling and simulation to help? Well, long-term, we'd like to use it to figure out which ESUS patients are most likely to have a recurrent stroke, but we don't have computational tools that are suitable for that yet. We don't have hemodynamic modeling online just yet, but we're working on it. We'd also like to figure out which ESUS patients are most likely to have incident AFib, because it might change the course of treatment for them. They might get put on an anticoagulant or something like that. And within that problem, we're really concerned with the sort of mechanistic question of, if ESUS patients have so much fibrosis, why don't they have AFib? So we think there are two possibilities here. The first is that there's something fundamentally different about the fibrotic substrate. So the spatial pattern of the fibrosis, or the distribution of fibrosis within the atria of the ESUS patients, might be different. But the other possibility, which I'll tip my hand here and say this is what we favor, this is our hypothesis, is that the fibrotic substrate is identical between ESUS and AFib patients, but there's an absence of triggers. There's an absence of focal activity in those patients, and so they're kind of like a ticking time bomb waiting for those triggers to form somehow, become malignant, and engage the fibrotic substrate and perpetuate AFib. And we think simulations can be really helpful in clarifying this ambiguity. So we availed ourselves of MRI scans. So 10 MRI scans of ESUS patients were taken here at UW as part of that neurology paper. And then we supplemented that with an additional 35 from our collaborators in Coburg in Germany. And then we were able to compare those to 45 AFib LG-MRI scans. These are all pre-ablation scans for first-time ablation patients. And I would take this opportunity to add, the imaging program here that Nazem and others have put together and maintained is an absolute boon for a lab like ours. We are swimming in data, and it's just an unbelievably rich environment for someone like me to work in. And so it's enormous. For example, the papers that I showed you earlier from my time at Hopkins, we were struggling to get 10, 20 MRI scans of AFib patients. And so the fact that we have hundreds banked from previous procedures here is pretty remarkable, and it makes my life really easy. Well, not easy, but exciting, let's say. So we developed, we adapted the existing modeling framework, which I already told you about. This is left atrial only model reconstruction, and we did that both because that's the chamber of most concern when we're talking about stroke, and also with the tools that we have here, we have automated, not automated, but we have predefined segmentations of the left atrium from the images. We don't get raw images, we can, but the raw images are sent to a third party, this company called Marisite in Utah, and they send back a pre-segmented mesh that we can use to really rapidly reconstruct a model. And what makes that exciting for me is that it's feasible to go from these segmented images to fully analyzed results in an order of a few hours, which compared to the type of simulations we were doing at Hopkins is basically light speed. And so what we want to answer is, in the ESUS patients with fibrotic atria, does the substrate stress test that we've already talked about show reentrant drivers? These slides are a little bit clunky because they're 90 individual images embedded here. And so we have 45 ESUS models, 45 AFibers. You can see there's sort of a diversity of the spatial distribution in the atrial geometry. In some of the models, both in the ESUS and AFib cohorts, pacing of the atria resulted in reentry. You can see we're pacing here from the right inferior pulmonary vein, and the driver is forming in the posterior inferior left atrium. I have to click again, wait, and here's a different model in which pacing simply never engaged the substrate. In this case, we paced from 15 different sites that were informed by mechanistic clinical data about the most likely sources of triggers in AFibers, and some models just were simply non-inducible. And so if we look at the split, interestingly, more ESUS or more AFibers as a proportion of the total population size were inducible to reentry. About 2/3 of them had reentry inducible, compared to about half in the context of ESUS. But remember, these ESUS patients had no arrhythmia symptoms whatsoever. They had no arrhythmias. They were followed for 30+ days with either Holters or Zio patches, whatever monitoring. And so this is an interesting finding. And what we really wanted to know here is, within those inducible models, was the fibrosis different? And so when we look at the statistics, we find that indeed they were not. There's no significant difference. In fact, the ESUS models had more fibrosis on average than the AFib models. And if we order these patients by the fibrosis burden, going from the least fibrotic to the most fibrotic, what we see is sort of the expected trend that having more fibrosis means there's more likely to be an inducible reentry driver. Although there were examples, and it's a little hard to see here, of models that were inducible to reentry despite having low fibrosis, and likewise there were some high fibrosis models that were not inducible despite the fact that they had all that fibrotic remodeling. And this is consistent with what we've seen previously, that there is, you know, shading and nuance in terms of what kind of fibrosis and the distribution of fibrosis matters in terms of having that special sauce, that recipe for initiation of reentry drivers. In terms of the arrhythmia dynamics, the number of pacing sites between ESUS and AFib for inducible models that actually induce a reentry, no difference. The number of distinct reentrant driver sites, so remember back to the original study that I was showing you, with phase singularities dancing, no difference. And so the implication is that the first hypothesis is not supported. We don't think there is something fundamentally different in our eyes, in our hands, from the standpoint of a computational modeler about the fibrotic substrate in these ESUS patients. So this supports by default the hypothesis. I don't think we've proven this yet, but it supports the hypothesis that there's a lack of triggers in these ESUS patients, and that's why they're not having AFib. So I'll reiterate that point. The absence of triggers hypothesis is supported, although we did see a higher prevalence of arrhythmogenic fibrotic substrate in the AFibers, and we think that that's to be expected as well. And if you're attending HRS, we have a poster about this, and you can meet Savannah and hear about all the nitty-gritty details and what she thinks about these findings. And plus, we'll probably have some new stuff by then. So hooray. So the last section of the talk is about optogenetic defibrillation. And so this is where I tell you all to buckle up, get ready. This is some Star Trek stuff. This is the real curb appeal part of the presentation. So what's optogenetics? Many of you have probably heard about this, especially in the context of neuroscience. Optogenetics is the genetic encoding of light-sensitive proteins called opsins in excitable cells, so that we can use light to evoke an electrophysiological response instead of electrical stimulation. The most commonly used opsin is channel rhodopsin 2. It's a light-gated, non-selective cation channel, and when you blast it with blue light, it elicits a large depolarizing current. It's not quite as large as the fast sodium current in a typical cardiomyocyte, but it's more than enough to trigger an action potential in a cardiomyocyte that's expressing channel rhodopsin. You see simulation data here, but of course this bears out, and this is all based on experimental data that we used to initially calibrate the model. So what are the big advantages of optogenetics? Different opsins exist. So channel rhodopsin is by far the most commonly used opsin, but there are also anion channel rhodopsins that can repolarize the cell rapidly. There are opsins that can mimic sympathetic tone upon illumination. There are many different ways that these can be used, which are super exciting and really, frankly, have been underexplored in the context of cardiac applications. It allows for contact-free perturbation, so you don't need to touch the cell physically in order to elicit an excitation, which is a big advantage for stimulation. Patterned illumination is a possibility. So there was a paper that showed that illuminating channel rhodopsin-rich cardiac monolayers with these pinwheel patterns could induce reentry spontaneously, and then illumination of the same rotor with the opposite chirality could dynamically reverse the flow of that, which is just exactly the kind of cool thing that an engineer like me gets excited about. But really, what we think is the main driver of potential applications for this, and what's made it a huge boon in the world of neuroscience, is the idea that you can selectively express the opsin of interest in specific cells or tissue. And so this is a hallmark of gene therapy. So you can attach a promoter to the transgene of interest and say, okay, I only want to see this in specific type of neurons in the brain, or I only want to see this in Purkinje cells in the heart, or I only want to see this in atrial cardiomyocytes. And these types of selective expression experiments have been part of the sort of emergent field of optogenetics and have made it really powerful. And the best example of this, I think, is to watch this mouse skitter around the big Rubbermaid container. This is a video from Carl Dijsselroth, sort of the father of optogenetics, along with Ed Boyden, who's now at MIT. But what's happening here is that there's an optogenetic probe, a blue light probe, that's been implanted in the mouse's brain, and it has channel rhodopsin selectively expressed in the motor cortex. And so when they turn the light on, all it wants to do is run around and turn left. So there you go. And you can imagine that with regards to things like deep brain stimulation, where implanting an electrode, you have off-target excitation of any number of other neurons other than the ones that the family of neurons that are being selectively targeted. And so this is what makes optogenetics so cool in the context of neuroscience. But what can we do in the heart? Okay, so the dawn of cardiac optogenetics came about 5 years after the big explosion of neuroscience optogenetics research. These are data from Philip Sass's lab at the University of Bonn, showing robust channel rhodopsin expression in mouse cardiomyocytes. These are transgenic mice, so all of the myocytes express channel rhodopsin. And this is an unpublished movie from the same study where they are focusing blue light through a macroscope on the Langendorf perfused mouse ventricles. You can see that they readily entrain excitation of the heart at a faster rate than sinus rhythm. So we saw this paper and thought, wow, we have an excellent set of computational tools for simulating cardiac electrophysiology and arrhythmia. And we know that the literature in optogenetics has detailed mathematical description of ion channel biophysics for these optogenetic constructs. Why not combine those and develop a comprehensive framework for simulating cardiac optogenetics so that we can see what we might be able to do? And that's exactly what we did. So at the protein scale, we have biophysically realistic opsin models. At the cell scale, we have representation of light-triggered action potentials in myocytes expressing different levels of channel rhodopsin or other opsins. At the tissue scale, we developed new methodologies to realistically pattern light-sensitive cells that would arise from gene or cell therapy to infuse or to inscribe light sensitivity into the tissue. And at the organ scale, and this is a really important point, we incorporated a model of light attenuation, because one of the things that we knew for sure, just from first principles, is that photons are absorbed and scattered by human tissue quite significantly. And so the effective penetration depth, especially for blue light and for ultraviolet light, is extremely low. It's less than a millimeter in terms of a space constant for the fall off of the effective irradiance in tissue. And so we knew we needed to implement a realistic model of light attenuation in order to have any idea of whether, for example, the heart could be defibrillated with light, the heart could be paced with light. Okay, so how can we use this? There are many applications, and if you're interested in this, I would point you towards our state-of-the-art review from a couple years ago. The in vitro applications, in fact, I would say more likely to succeed in the long term. So this is things like high throughput all optical screening to do drug cardiotoxicity assays. But I'm not going to talk about that today because it's not my favorite. My favorite is the idea that maybe someday we could defibrillate the heart with a gentle pulse of blue light instead of an aggressive electrical shock. And so we worked with Philip Sauce's lab at the University of Bonn to do side-by-side experiments and simulations. So Philip had a mouse model where he was able to induce stable and non-spontaneously terminating tachycardias in these Langendorf perfused hearts. And on our side of the ocean, we had a previous study simulated human heart. So this was a patient who had a huge infarct with extensive border zone surrounding. You can see that in gray. And this was from a previous study at Hopkins. So we decided to see, like, well, if we express channelrhodopsin in the human heart, or if we simulated the expression of channelrhodopsin in that particular heart, could we defibrillate the tachycardia that emerges in the simulation? Okay, so Philip did an enormous number of simulations here and found robust termination of the ventricular tachycardias in the mouse models. I'm really underselling the experimental data here. There's a ton of different permutations in the paper itself, different types of arrhythmia models. They did in vivo transfection as opposed to transgenic animals as well. And across the board, the finding was it works. That we're able to reliably terminate VTs in these mouse hearts with light. Okay, so that's good news. What about in the human heart? So this is at baseline, the tachycardia induced in the model. There's a pseudo electrocardiogram down here in the bottom. Keep losing my pointer. Oh well. And so we applied a blue light pulse. So this is the simulated channelrhodopsin expressing ventricles. Lo and behold, it didn't work. Okay, so what happened? Why? What's going wrong? Well, we knew light attenuation was going to be an issue. We anticipated that. That's part of the reason why it was included in our model. And we thought maybe the issue is that the optogenetic stimulus, which is being applied at the epicardium, you can see the halo around the ventricles here of blue light. Maybe it's just not penetrating deep enough. Maybe it's not having any action on the subendocardial layer of myocardial tissue, and the reentry is not being interrupted. And so at this time, there were new papers coming out at the rate of like 1 a month of different channelrhodopsin variants that were molecularly engineered to respond to red light instead of blue light. Red light penetrates about 3 times deeper in cardiac tissue than blue light, and so we thought this was a potential solution. And so we stimulated with red light at the epicardium, and we saw, sure enough, whoa, tachycardia terminated. Excellent. That's quiescent here. Of course, that's because there's no sinus node in this model. And so the person wouldn't, I assure you the person wouldn't flatline, theoretically, this virtual human. So why? Why did this work? And this is where models come in. So you see that the surface has been cut away here, but in the model itself, in the simulation itself, that surface is still there. But we're just able to peel back the layers effortlessly and look at exactly what's happening in every single region of the model. What we see is that when the blue light is applied, we're looking sort of side by side at the same time points, exact same time points for blue versus red. We see that for the blue light case, propagation of the reentrant wavefront into the right ventricular free wall is successful because of incomplete depolarization of the wall. Okay. In contrast, you see that the extent of depolarization from the light stimulus in the red light case is so significant, it's basically transmural, that the propagation of the reentrant wavefront into the RV free wall fails, and we end up seeing a completely different wave form manifest. This is in the death throes of the tachycardia, and ultimately, while the blue light case continues unabated, the red light case has a termination of the arrhythmia. Cool, okay. And we were able to dig even deeper, looking at individual node levels, looking at the level of sodium channel inactivation, and show mechanistically that the reason why red light worked, blue light didn't, was fundamentally because of the fact that it blocked sodium channels. It raised the membrane potential high enough and did so in a sustained manner that even though there was still propagating activity in the optogenetically depolarized tissue that was predominantly driven by calcium depolarization, it was simply too weak for the reentry to be sustained, and interrupted propagation and terminated the VT. So what have we learned from here? Well, we think optogenetics could someday work for defibrillation. But if it does, overcoming the light attenuation problem is going to be a hugely important constraint. We think that computational models can help guide new experiments. And specifically, at this point in the field, there were big considerations being given to, doing larger animal optogenetic models, doing rabbits, pigs, dogs. And what our work suggests, and this was, our collaborator Philip told us this specifically, like, well, we're not going to do those now. We're gonna wait until we know that we can robustly express red light sensitive opsins in these larger animals, because we don't want to waste the animals, and we think there's an ethical consideration. And so this work really does, even though it's kind of pie in the sky and nerdy engineering stuff, it really has changed the course of how the experiments have been done in this field. But I do want to emphasize that enormous barriers to translation still exist. Just the idea of letting someone do gene therapy to light sensitize your heart is completely crazy right now, and no one should ever consider it until the safety and efficacy and long-term feasibility, like, it has to be a huge open and shut case that yes, this is way better than getting an ICD in order for this to progress. But if and when that does happen, if and when we do find safe ways to do it, we know that we'll have these computational tools that will help us make sure it's the best light-based defibrillator possible. Okay, so that's the end of my talk. I want to thank my lab. Kelly, no, Kelly's not here, but she drew this wonderful cartoon. Chelsea's so new that she doesn't have a cartoon, so please imagine that as a cartoon. I want to acknowledge the contributions of my collaborators, especially the electrophysiology team here at UW, who've welcomed me with incredibly open arms, and of course our collaborators in Coburg. Thank you.
So you are introducing patient data from MRI. What about other ECG has a lot of data that's different. Where do you change how the monitors are based on something?
Yes. So the question. Well, I guess you said it into the mic, so I don't need to repeat it. Okay. Really nailing it. Yes, so we only personalize the models currently with information from MRI, so it's structural information. We do customize the different regions, of course. So like I said, in the atrial section, the fibrotic action potentials are different from the non-fibrotic action potentials, but they're an average human model. Yes, we are working and thinking about ways that we could personalize the electrophysiology, but it would have to be, in order to be feasible in clinical workflows, we think it would have to be non-invasive. So electrocardiographic imaging, for example, so ECGI, that's electrocardiographic imaging, the idea of putting a vest on a patient, and it's kind of like an ECG on steroids, where you take a CT of the torso with all the electrode positions and a CT of the heart, and then you solve the inverse problem to reconstruct a map of excitation and repolarization times on the cardiac surface. So we think that that type of information that has spatial heterogeneity could give us some information about patient-specific activation recovery intervals and conduction velocities, but we're not there yet. Yeah.
Of course, Jordan from electrophysiology team.
Number one is all your models are the 2D structure there, and we know like there's very different activity from raw tongue looking at VT and the circuits that are involved in that. So how do you get to that point where you're looking at this more in a 3D model in terms of the cell layers? And the second question has to do with conduction velocities, I guess. You talked about trying to look at the anisotropic conduction related to the muscle fiber orientation, but do you get into concerns or issues looking at like connection density? So especially when you're looking at patchy regions of scar, how do you model propagation velocity through that?
Okay, so I'm supposed to repeat the question. So the first question was, okay, so 2D versus 3D. In fact, most of the model data that I showed today were in volumetric 3D measures. The new atrial simulations that I'm doing here in UW, we've very specifically gone to a quasi 2D methodology because it's faster and easier to do. But even those atrial simulations, they're not a complete 2D representation or like a manifold representation. What we actually have is an endocardial layer that's coupled to an epicardial layer, and we can modulate the connections between the epicardium and the endocardium by taking out the sort of the linear connections between epi and endo. But yeah, it's definitely an important question. And of course, you know, in the VT data that I showed for the optogenetics, there's absolutely transmural heterogeneity that makes a difference in the manifestation of the particular reentry drivers. The second question was about conduction velocity. Yeah, we have a devil of a time explicitly representing connexin because the mathematical formulation for representing reaction diffusion in these models homogenizes the entire volume. And so we essentially have to say, okay, we can incorporate spatially heterogeneous anisotropy according to the local orientation of fibers. But if someone sends us data that say like, oh, okay, well, we have a 20% downregulation of connexin 43 expression, there's no really good way of explicitly incorporating that in our model. And so thus far, the approach in the field has been to try and take macroscopic measurements of conduction velocity and use that to calibrate the conductivity. But yeah, it's super hard. Just measure the conduction velocity. So pace, see an ellipsoid that spreads out, you know, at 60 centimeters per second along the fiber orientations, and 20 centimeters per second transverse to the fiber orientations, and calibrate the model accordingly. But it's maybe the Achilles heel of this type of modeling is the lack of explicit representation of intercellular coupling. And other groups have done that type of model where they have kind of a brickwork of cells, or of simulated cells with actual explicit representation of intracellular medium and connections. But at scale, it would be impossible to incorporate those and simulate the expectation of the entire part. Yeah, so that's an excellent question. And so I'll repeat it for the Zoom viewers. The question was about the progression of the evolution over time of the fibrotic substrate in the left atrium, which is of course dynamic. Yes, we're thinking about that. So there are a couple different ways that we can do it. We don't have a lot of longitudinal data where we have sequential MRI scans from the same patient over and over again to look at how their fibrotic substrate changes. But we've thought about potential applications where we could algorithmically grow. Like if we could take one scan at time 0 and then take a second scan a year later and develop a model to represent, okay, well, how does it get from A to B? Then that's something that we think is feasible. The second thing that we've been thinking about, and Fima Mashere is here in the back row, he's a cardiology fellow, and he's going to be working on a project with me and Nazem that looks at models that combine pre-ablation MRI scans with post-ablation MRI scans, because the contrast on the post-ablation MRI scan jumps out really, really bright. We can see the exact locations, or we hope we can see the exact locations where the ablation is actually taken. And we think that there's a lot to be learned about potential for recurrence when we take both of those sets of scans, merge them into the same geometry, and then run simulations on models reconstructed from that. With regards to the first comment, which was about the sort of integration of this bioengineering team with the clinical collaborators, let me give a sales pitch here. Bioengineering is a dynamic substrate. Bioengineering is changing. Sorry, I got a cold. My personal belief is that we are at the onset of the next phase of bioengineering. And phase one was build better doohickeys. Use engineering principles to build better things that you can use in the clinic. Better gantries for the MRI scanner. Better this. Better scalpels. The second phase was use bioengineering approaches, use engineering method to design better experiments. Tissue engineering, cell therapy, this type of thing. The next phase, and I like to think that we're at the vanguard of that, is integrating in the clinical workflow, is bringing bioengineering principles to the bedside and doing so in a way, my lab comes over here every Tuesday morning to meet with the cardiac EP team and to attend the meetings that the fellows go to. We want to be immersed in the hospital, and UW is doing an outstanding job at that, in my opinion. Not necessarily just in the bioengineering aspect, but in the fact that the hospital is right here, which is something that a lot of programs don't have the privilege of. Okay, so the question was about novel. Right. Right, absolutely. Yeah, so the question was about simulating novel arrhythmia models, and absolutely, I think the paper that you're talking about is the one in Scientific Reports, which was the comprehensive framework for HFPSC-CM injection. Yeah, so this is something that's a going concern, and I didn't talk about it here, but what we rely on in order to thrive in those modeling playgrounds, we're not a lab that designs cardiomyocyte models. We take cardiomyocytes models from others, we implement them, and then we simulate their effects at scale. And so there's a deluge now of new mathematical modeling and computational work in the area of HFPSC-CMs that's enabling us to do that. And the main rate limiting factor, if we wanted to simulate some novel cardiomyopathy in dogs or rats, animals, some animal model of arrhythmia, is do we have access to a suitable action potential model? And do we have enough data to inform the model? So a few biomarkers is usually enough. Conduction velocity, upstroke velocity, action potential duration, disease and normal. That's usually enough to give us a really good starting point.
One last
Well, okay, so the question is, how did the 10 patients in the OPTIMA pilot study do with respect to historical controls? If you read the paper, it would suggest that they did very well, because the rate of recurrence of persistent AFib, which is the disease that was being treated, or the condition that was being treated, was very low. None of them recurred to persistent AFib, which isn't always the case if they come in and get a garden variety pulmonary vein isolation. And I think that the likelihood of success at 1 year for a PVI in persistent AFib is on the order of 50 to 60%. I'm looking at Ann and Jordan here. That's reasonable. Not all of those patients were followed up on out to a year, though. And so we're not sure. And I'm not personally comfortable doing an apples to apples comparison with the pilot data set that we have. I'm not convinced yet. I'm a skeptic. But I think we're optimistic. And the ultimate test will be the randomized controlled trial. And I'll tell you, the endpoints for success in that trial are stringent. They're looking 30 seconds of AFib is being classified after the 3-month blanking interval is being classified as a failure. And so, yeah, we're the key is recruiting the right patients. But yeah, it's very exciting. Yeah.
Thank you very much, Kat. It was a great talk.